Assalamualaikum warahmatullahi wabarakatuh. We are group 5. I am Muhammad Arman Syabil bin Jazmiaizan. So today, my group was given the task of conducting the experiment of DNA extraction.
Assalamualaikum warahmatullahi wabarakatuh. My name is Mohd Jazli bin Juhairis.
And I am Zulaikha binti Azman.
And I'm Farhana binti Shukri. So I will do the experiment while other members will conduct by giving the instruction to me. So let's get started. The learning outcome of this experiment are: first, to extract DNA from banana using the material that available at home. Second, to identify the presence of the extracted DNA from banana. The material that we use in this experiment are banana, 110 ml filter water, 20 ml liquid dishwasher, 20 ml hand sanitizer to replace the alcohol, glasses and strainer, plastic ziploc bag, salt, and lastly, knife and cutting board. So let's do the experiment.
Put 20 ml liquid dishwasher into the 110 ml filter water.
For the second step, add 1 teaspoon of salt in the mixture and stir it.
Cut the banana and peel the skin off and then put it in the ziploc bag.
After that, don't forget to put the mixture solution with the banana together.
For the fifth step, clip the ziplock and smash the banana inside until all the lamps are gone and it looks like pudding.
Get a glass and strainer and strain the mixture into the glass.
Get your 20 ml cold hand sanitizer from the refrigerator and add it to the mixture. In this experiment, we have found out that DNA strand can be extracted from fruits and vegetables. DNA is a genetic material which all living organisms have and function to pass on information from one generation to the next. DNA information also tells our body how to work and grow and develop. So from this experiment, we have used banana as a sample. This is because banana is easy to be obtained and will give a clear result. Banana also can be replaced by strawberries and kiwis because all of it works well for this experiment. To extract the DNA from the banana, it first must be mashed as much as we can but avoid making too much bubbles. This is because bubbles will make the result to be not clear as it can be. So the banana is then mixed with salt because salt will help the sample to be prepared before it being added into liquid dishwashing detergent, which is this soap. When the dish soap helps release the DNA, this salt will help the DNA strand to stick to each other in clumps large enough for us to see. From the experiment, we can see that this soap can help split apart the membrane, the outer skin that holds cells together. This membrane are made of a type of molecule called lipid, and lipid is known as a class of organic compounds that are fatty acid, which is insoluble in water but soluble in organic solvent. They include many natural oils, waxes and steroids. By splitting up this membrane, the DNA can be released. So next, the alcohol is added to help to clump the DNA to form. This happens because DNA are soluble in some liquid but not in alcohol. This is why alcohol will help the DNA extracted to clump. When adding the alcohol to the solution, it must be added slowly to allow the alcohol to form a layer on top of the solution. the banana solution prepared is also not supposed to be heated in the water bath for too long to avoid the DNA of the banana from breaking down. We also should avoid from using a strong type of dish soap because it may cause the membrane to break down rather than splitting it. This will probably make this experiment to fail because the DNA cannot or fail to precipitate out from the solution. So last but not least, we should not shake the solution after adding the alcohol because this would make the solution and the alcohol to mix.
Okay, for the conclusion about this experiment, I can say one of the method in the extraction of DNA of banana is by using hand sanitizer, dishwashing liquid and salt. Then when dishwashing liquid was applied to the cell of banana, it can capture lipid and protein. Using this method, the DNA of banana can be isolated as it forms clumps of DNA in the solution. So thus using this method, we can identify the presence of DNA of banana. Okay. So that's all from our group. Thank you.
Thank you.
Bye.